Hey guys, this video is about the cell cycle, which is all the steps that the cell goes through as it goes from parent cell and the creation of two daughter cells. And what I need you to do is grab a piece of paper or your whiteboard and draw out this circle. You can leave all of this kind of stuff out in here, but you definitely want to include this pie taken out and get this drawn. And what I want you to do first before you watch any of the videos, I want you to try to label as much as possible. So obviously we know that the two major steps of cell division are cell division, or the M phase, and we have interphase. But beyond that, I want you to try to label all the phases, right? So we have three phases here to label. Try to label them and describe what's happening. We have two major phases that happen during cell division. Label them and describe what's happening. And also, there are three checkpoints. I want you to try to write down what happens in each checkpoint. So go and try to label it first on your own, then unpause the video and watch the rest. All right, so hopefully you guys have this labeled and you've tried it. So let's go through this, guys. All right, so in the very first phase, we have G1, which is called gap 1 or growth 1. And the key thing that happens here, guys, is that the cell is growing and getting larger. Now, during this time, during growth 1 or gap 1, the cell is doing all of its normal activities. So, for example, if it's a nerve cell, it's transmitting signals because that's what nerve cells do, okay? But the big difference that we're going to see here from the beginning to the end, here we have our normal cell starting off and has a nucleus in it. By the end, it is a larger cell, still just one nucleus in it, okay? At the end of G1, we have our first checkpoint called the G1 checkpoint. And here it's checking for proper growth. Is it big enough? Do we have enough nutrients? And has the DNA been damaged? If it's not meeting any of these, it's going to stay in G1 or enter something called G0, or it could actually destroy itself. The cell would rather destroy itself than replicate incorrectly, because incorrect replication leads to things like cancer. All right, let's assuming everything was good. It passed the G1 checkpoint. It's then going to go into the S phase. S stands for synthesis, or to make, and it's making DNA. It's going to replicate all of its DNA. So at the end of synthesis, we still have our one cell, and we still have our nucleus. But let's take a look what happened, guys. We went from one strand of DNA, and now on the inside of the nucleus, we're going to have two strands of duplicate DNA. All right, the next phase is called G2. And the important thing that happens in G2 phase is that organelles get synthesized. Organelles need to be synthesized because each daughter cell needs to have the things it needs to survive. It needs to have an ER. Each cell needs to have mitochondria. Each cell needs to have a Golgi, right? So it's synthesizing the organelles here. At the end of G2, there's another checkpoint checking for cell size and then making sure the DNA got replicated correctly and there's no damage. Again, it would rather destroy itself than keep going. All right, so interphase is the getting ready part. It spends most of its life during interphase. 90% of the life cycle is in interphase. And during this time, the DNA is in the form of chromatin, okay? Now it's going to enter into cell division. The chromatin turns into a chromosome, okay? And two things happen. One is mitosis, and the key thing about mitosis is that it's the splitting of the nucleus. So basically what we have here, we have our cell, okay? Here's our cell. And we have our nucleus. Here's how we start. It has two pieces of DNA. So that's the starting. But to end mitosis, we have our cell. And we have two nuclei. Okay? So that's what happens in mitosis, the splitting of the nucleus. During mitosis, there's also a checkpoint that takes place. It's just making sure that mitosis is taking place correctly. Okay. So at this point, we have not split the cell yet, okay? We have just one cell, okay, again, and we have two nuclei in that cell. Each of them have some DNA in them, have the correct amount of DNA. To finish this, we're going to do a process called cytokinesis. Cytokinesis splits the cytoplasm, and we take one cell with two nuclei, and we're going to finish and make two cells, each with their own nucleus. Each one can then re-enter into the cell cycle and reproduce and split. All right, so if you guys had trouble with any of this, if you have trouble labeling this, you definitely want to try this again with a fresh sheet of paper.